Hello, everyone, and welcome to today's webinar, Supporting Drug Development for Advanced Therapies with High-Quality RNA Analysis, the Critical Role of Sampling and Pre-Analytics. My name is Delia Osborne with Labroots, and I will be your moderator for today's event. Today's educational webinar is presented by Labroots and brought to you by Pre-Analytics. To learn more, visit preanalytics.com. We encourage you to participate today by submitting any questions that you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of today's presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speakers for today, Dr. Björn Lundin, Burmeister, Project Manager, Tata Biocenter, and Dr. Kalle Günther, Director, R&D Pre-Analytics. Dr. Günther, you may now begin your portion of the presentation. And please stand by. We are experiencing a small technical issue. One moment, please. We appreciate your patience. Again, we appreciate your patience as we try to troubleshoot our connection and ensure that we can bring you a presentation today. So please stand by, and thank you again for your patience.
Thank you very much for the introduction. I'm giving this presentation within my role as a developer of the Paxton blood RNA system and other products of pre-analytics. And today we will learn more about how standardization of the pre-analytical steps from blood specimen collection to isolation of RNA helps to get reliable and repeatable results from blood RNA analysis. First of all, who we are, what is our mission? This slide covers this, and important to mention here is that we develop, manufacture, and sell integrated and standardized systems for sample collection, for human sample collection, and for different specimen types and different analyte classes. This is our legal disclaimer for this presentation, and for further information, please have a look into the manuals of the products that we mention. So why are pre-analytical errors and in workflow so important for diagnostics? So not only for diagnostics, but also research, biobanks, and other projects. So the answer can be given with some numbers I'm presenting here. First of all, there's a report published in 2015, and it was found that 10% of all patient deaths and about 6 to 70% of adverse events are caused by diagnostic errors. The pre-analytical phase accounts for 46% to 68% of such errors observed during the total testing process, and that was published in the Medical Laboratory Observer in 2014. So this means that the pre-analytical phase of the diagnostic workflow is the biggest contributor to diagnostic errors. These are quite high numbers and high damages to patients, also to the healthcare system and research. And where do these errors come from? They are caused by pre-analytical influences on the sample, lack of standardization of pre-analytical steps, and the reference material used. What happens to blood specimens after collection into commonly used blood collection tubes, especially in EDTA tubes? In this figure, the impact of blood storage time on FOS gene expression in 3 different donors with blood collected into multiple EDTA tubes per donor is shown. RNA was isolated from EDTA blood immediately after blood collection and after storage for 15 minutes, 30 minutes, 60 minutes, 1 day, 2 days, and 3 days. in order to measure the fold changes of phospho-mRNA levels. In the figure, we see a baseline test time point 0 corresponding to the in vivo phosphotranscript level of 100% or 1-fold with a red line, and this marks the original level in the donor at time of blood collection. The phos is upregulated in EDTA whole blood just within 15 minutes. It reaches its maximum after 1 day and starts to decrease afterwards. At each test time point, the phospho-mRNA level measured does not reflect the original level as it was in the donor's body at time of sample collection. Changes are the results of two main principles. First of all, the vital cells come from a living organism, and they are transferred to a collection device, which has a total artificial environment. Cells come into contact with glass, plastic, rubber, the oxygen, nutrition supply is different, temperature decreases, and for the cells this is a crisis, and they react to this situation in this artificial environment quite fast. For example, they change the gene expression, so we see gene up and down regulation. For phos, this transcript that we focus on here, that is the upregulation. They also change the protein profiles. There are modifications of proteins and changes of metabolite profiles and other changes. The living cells continue to change the sample as long as they live, during transportation, during storage, until they arrive at the laboratory. But even in the laboratory, with additional storage time for batching multiple samples for a sample preparation run, and with the first analyte extraction steps, the sample can still change until it is lysed and all the vital processes are stopped. So the degree of changes is subject to donor-specific response. And we can see this here. These are 3 different donors, and you see different degrees of responses. And we meanwhile know also from other genes that we analyze that those can behave differently. So for example, they show even stronger changes into both directions. Gene induction leading to higher transcript levels, but also gene repressions leading to substantial losses of transcripts. The second principle is RNA degradation. So the undersupplied cells die and the intracellular RNA is degraded in this process, leading to RNA fragmentation and loss of RNA integrity, resulting in loss of RNA target sequences. The conclusion of this experiment is that the period of storage time determined the level of phos transcripts quantified, and that the measured levels are not the ones originally existing in the donors. This makes evident that blood stabilization at time of collection is key for reliable gene expression analysis, especially if we do not know how the genes we are targeting behave in our workflow setting. But let's come to the overview of the entire workflow of gene expression analysis from blood. So it starts with the donor or the patient. It continues with the specimen collection and nucleic acid profile stabilization, followed by transport and storage, and the nucleic isolation and storage procedure itself. And this forms the pre-analytical workflow. And after the pre-analytical workflow, we have the analytical part of the workflow. That basically means the downstream assay, the analysis of the RNA by, let's say, RT-PCR, could be RNA-seq, NGS, also droplet PCR as well. And then the data is analyzed and reported back to the physician or the patient. We need to understand that all pre-analytical steps can have an impact on RNA profiles, quantity and quality of RNA, and that the molecular assay cannot discriminate anymore the changes happened to the sample before. This can lead to a wrong diagnostic result because an artificial gene expression profile is generated due to the selected pre-analytical steps of the workflow. Therefore, all pre-analytical steps should be standardized and meet the pre-analytical requirements defined in the EU in vitro diagnostic regulation for workflows in Europe. And also the FDA recognized the importance of the pre-analytical phase by providing the guidance for assay validation to encompass the entire workflow from sample collection to assay output. It's very important to consider and understand this, and that the analytical test result is always a result of an entire workflow, and the pre-analytical part of the workflow needs to be standardized. To standardize the pre-analytical phase, we developed the packaged blood RNA tube and kit. And the tube is shown on the upper left part here. It's a plastic tube evacuated to enable a blood draw of 2.5 ml blood. It contains a liquid formulation of an RNA stabilization additive. And the tube is linked to a packaged blood RNA kit. The packaged blood RNA kit is based on proven silica membrane technology to isolate RNA, and we included a DNA removal step by performing a DNA treatment on the spin column membrane that removes traces of DNA that gets otherwise co-purified with the RNA, and then the RNA is diluted with a proprietary elution buffer that enables full performance of the RNA in later downstream assays. And both parts are combined and form the packaged blood RNA system. And the system helps to standardize blood collection, stabilization, transportation, storage, and isolation of highly pure and intact intracellular RNA to avoid pre-analytical errors in accordance with ISO 21861. The system was CE certified and received 510K FDA approval in 2005, and it is still the only FDA-cleared system for blood RNA in the market. Since its introduction in 2001 as a research use product, several products have validated their workflows with the system and point-of-care testings with the packaged blood RNA tube. And there are more than 6,000 scientific publications available that describe studies using packaged blood RNA, and the number of publications is still increasing. I would share with you some technical details, what happens during blood collection in the packaged blood RNA tube. So as soon as the blood tube is connected to the patient, blood gets into contact with the RNA stabilization reagent in the tube. So the cells are immediately lysed in the tube by strong cationic detergent of the stabilizer. So there are no intact cells for separation or enrichment of certain blood cell populations in the lysate anymore. And the detergent binds to the nucleic acids, also to proteins and other biomolecules. And by this, the enzymes are inactivated, resulting in two effects. So first of all, there is no RNase activity anymore. And the second effect is that there is no active transcription machinery anymore. And then we see additional effects. So the RNA is protected by binding to the cationic detergent. So this detergent is positively charged and it binds to the molecules, and the hydration shell gets stripped off and leads to the precipitation of RNA and other biomolecules, such as DNA, proteins. And this takes 2 hours at room temperature, and the precipitates can later be harvested by centrifugation. And by the centrifugation, we have a pellet at the bottom of the tube here, and that is the starting material for the RNA isolation with the packaged blood RNA kit. I would like to show you some performance verification data of the packaged blood RNA system in a time course experiment. Here, blood samples from 10 donors were collected and stored up to 3 days at room temperature. And at each test time point, duplicate samples per donor were subjected to RNA isolation with the packaged blood RNA kit. And after the last test time point, so after 3 days, the transcript levels of FOS and interleukin-1 beta were analyzed with duplex real-time RT-PCR assays. And the changes of transcript levels were calculated with delta-delta CTs. And as you can see, that all delta-delta CTs of the packaging samples stayed within the method's precision of both assays. So the method's positions are indicated with red dashed lines here, and each individual data point is within this range of acceptable data. For PAX-gene, while unstabilized samples showed significant transcript level changes. So in case of FOS, we see this induction indicated by negative delta-delta CT values. And for interleukin-1 beta, we see a different behavior. We see induction, negative delta-delta CT values, but also positive delta-delta CT values. This means loss of transcripts during period of storage. We did not only look into short-term storage, so storage for days at room temperature, but also in a similar study, we verified the performance of the PAX-gene blood RNA system with blood samples that were stored for up to 11 years. So we called it long-term storage, and we used the commonly used freezing temperature of -20 degrees Celsius here on the left side, and on the right side, we applied deep freezing storage temperature of -70 degrees. And again, we collected multiple blood samples from 10 donors. The samples were frozen up to 11 years. And at each test time point, duplicate samples were processed. And the RNA was analyzed with FOS and interleukin-1 beta. And shown here are the delta-delta CT mean values between stored and unstored samples. And what we can see here is that all mean values stay within the method's precision. So this confirms that is only a subset of data. We have more. We looked also into RNA integrity, yield, purity, and so on. But that is important to see that the transcript levels are not, in fact, impacted by long-term storage at those freezing conditions. So 11 years was the endpoint of this study, but we meanwhile have evidence and published also with a scientific poster that blood samples are stabilized for up to 17 years of storage at -80 degrees. That is especially important for biobanking purposes. This slide gives an overview of the PAX-gene blood RNA workflow and some of the verified performance claims. We discussed already here storage at room temperature for up to 3 days is verified. At refrigerated conditions, we verified storage of up to 5 days, 11 years of storage at deep freezing temperatures. We also verified that samples can withstand 2 freeze-thaw cycles, because it can often happen that samples are thawed during transportation and then they got frozen again in the incoming lab, and for processing they are thawed again. But this does not impact RNA quality, quantity, and there's also no impact on transcript levels. That was thoroughly verified. And not on this slide here, but this is also verified. We guarantee that at least 95% of samples provide at least 3 microgram of total RNA. But the typical yields are much higher. So they are about 9 microgram per tube or per 2.5 ml of blood. Pre-analytics offers multiple options of RNA isolation from one PAX-gene blood RNA tube. So the different options address different regulatory requirements of IVD and research use application. Sample throughputs are addressed. We have manual and automated procedures, and also the potential need of customers for co-isolation of small RNAs, such as microRNAs. The automated procedures are available for QIAcube and QIAsymphony instruments, and we use trusted spin column, silica membrane, and magnetic bead technologies. That is my part for today, and I would now like to hand over to Björn Burmeister from Tata Biocenter, who will showcase the use of the PAX-gene blood RNA system with patient samples.
Hello, and thank you, Kalle, for the first part of the webinar. My name is Björn, and I'm a project manager at Tata Biocenter. And in this session, I will take you through how we, as a CRO, we support the development of advanced therapeutic drugs. and we go through everything from handling and extraction to method development and clinical sample analysis. And as you know, blood is the most widely used sample type in bioanalysis, especially when evaluating how a drug is distributed and how safe it is. This becomes even more important in later stage clinical studies, where blood analysis plays a key role in guiding the development of life-changing therapies. And our role as a CRO is to ensure that from the moment a sample is received, its biological integrity is preserved, and that the data that we generate truly reflects what is happening in the patient. And we do this by focusing on sample handling and workflow, method development and validation, and the importance of quality in clinical analysis. So let's begin by looking at the first step in this process, where everything starts, the sample itself. And now this workflow may be familiar to some of you, but for others it might become as a surprise, especially when you see the number of quality control steps that we have implemented to ensure that we deliver the highest possible standard. So in drug development, every step from sample collection to the final analysis is critical. And why is that? It's because we want to preserve the phenotype from the patient. This is very critical. Sample and analysis should represent the patient phenotype. And we focus on biomarkers, which are molecular indicators of disease progression or treatment response. And the key to trustworthy biomarker analysis is having full control of the sample journey. And as you can see, Tata Biocenter is located in Sweden, in Gothenburg, and we get samples all over the world. And we manage this through controlled shipping, with including temperature loggers. And this allows us to verify that the sample remains within the required temperature range during the transit. And this protects integrity from the moment they leave the collection site. We have meticulous registration and storage and temperature monitored access control rooms. And this not only maintains sample stability, but also ensures full traceability and compliance with regulatory standards. We also have backup systems such as UPS and generators, and this ensures that critical equipment such as freezers and analytic instruments remain operational during power outages. This is preventing the loss of valuable samples and protecting ongoing analysis from being compromised. We also have redundant lab capacities with automated extraction robots and validated instruments. And by having these parallel systems in place, we avoid delays and maintain consistent turnaround times, while the automation helps reduce variability and increases the throughput. And one of the most important things is that we have highly skilled personnel at every stage. And that ensures that protocols are followed, that deviations are caught early, and results are reliable and scientifically sound. And of course, we have implemented quality control at every step. That means that from the sample receipt to the data reporting, we apply rigorous quality checks to ensure accuracy, reproducibility, and compliance. And this whole structure is very essential to ensure that the data quality and reproducibility, especially when you're handling clinical samples. Something we understood early on is that what happens before analysis, which is the pre-analytic phase, that has a huge impact on the results. And we at Tata, we were early to recognize this. And the publication on the right was an initiative together with Media already a decade ago. It highlighted the importance of best practices, which developed into an international guideline for molecular diagnostics. On the left are some bullet points for our findings. And pre-analysis, as Kalle introduced, includes how samples are collected, stored, and transported. And these steps can affect the transcription levels. And sometimes this is has even a more impact than the biology of the disease itself. And over the last decade, we have compared storage solutions, we have tested stability, optimized protocols, including the packed in blood RNA tubes usage, which stabilize RNA immediately after blood collection. Our contribution has helped to define how to maintain transcriptomical integrity from the moment of sampling, which is fundamental for everything we do in the downstream analysis. So once the sample is secure, the next focus is to develop and validate methods for the analysis. And we develop custom assays with ultra-high sensitivity, which are able to take just a few copies of target genetic molecules, which are often used for biodistribution or pharmacokinetic studies of oligonucleotide drugs. We also analyze changes in gene expression to monitor target engagement, off-target effects, or therapeutic responses. And this table showcases a typical of our validation process for qPCR and dPCR methods. So what we first start with is a feasibility assessment, and that determines if the current assay or method is suitable to move forward with. The next step is the method development, where we work to optimize the protocol so it's ready for qualification or full validation. And then, depending on your acceptance criteria or regulatory needs, we either can go for a method qualification or a method validation. And by following this systematic workflow, we ensure a logical progression that maximizes efficiency and reduces the risk of overlooking critical steps of having to repeat processes. This approach supports robust assay development, reliable analysis. So a few examples of what we analyze in this process. We look at the extraction efficiency, and this ensures that the method consistently recovers sufficient amount of the target from the sample extraction. We also look at assay efficiency and amplicon confirmation, and this is to verify that the assay amplifies the target with high efficiency and specificity, confirming that the right product is being detected. We also assess linearity, precision, and accuracy, and this is to demonstrate that they deliver reliable, reproducible, and quantitative results across a range of concentrations. We also look at matrix effects, and this is to identify and control for substances in complex matrix like blood that could interfere with amplification and skew results. And all of these steps ensures that the method works reliable, even under real-world conditions. And most important, it's not just about passing the QC metric. It's about building the confidence in the results so that decisions can be made based on them, whether that's advancing a drug candidate or monitoring clinical response. So a great example of this is our approach, is our automated packaging workflow. So in this workflow, we are using one of our key Symphony systems for RNA extraction. And consistency is the key here. So what you are seeing on this slide are two bar charts representing around 50 patient samples per batch, and each bar shows data generated by different scientists across different fronts. And what is very important to highlight is that every sample, whether it's the first or the 500th sample, must be treated with the same level of care and precision. In studies involving hundreds of samples, it's critical that we apply the exact same workflow, quality checks, and handling procedures to every single one, regardless of who is processing it or when. And that's how we ensure reliable, reproducible results across the entire study. On the left is the graph that represents RNA quality number, RQN, which is a general indicator of total RNA integrity that ranges from 1 to 10, with higher values indicating a better RNA quality. And usually values above 8 are regarded as high-quality RNA. And as you can see, the batch RQN, or the average of the batch RQN, is above 8 for these samples, which not only confers good RNA quality, but also reflects the consistency of our sampling handling process. And on the right represents the RNA yield, which most samples yielding about 3 micrograms of total RNA, which is the expected output from the packaging extraction method, and also sufficient for downstream applications such as gene expression analysis or sequencing. So the takeaway from this, that with our validated automated process, we can get reproducible RNA quality and yield independently of the batch check. This is essential for reliable downstream gene expression analysis. So why does all of this matter? It's because your gene expression data is only as reliable as the RNA is based on. So let's take a closer look at the graphs on this slide that represent data over several hundred of patient samples. The upper graph shows results from a typical reference gene panel, where we measure the average expression of these genes across different RQN intervals and then normalized all values to the group with the highest RNA integrity. To summarize this, on the right, we have the average standard deviation and the CV. And what we found is quite striking. So just by dropping one RQN level led to an average of 25% decrease in the measured gene expression. Even more importantly, not all genes behave the same. Some drop more, others less. And this tells us two key things. First, that RNA degradation clearly impacts measured expression levels. And second, it underscores the importance of carefully selecting and qualifying your reference gene, both in terms of number and biological relevance, and to your specific application. The second graph shows gene expression for selected information-related transcripts using the same normalization approach as the graph above. And here too you can see that the variability in how different genes is responded as the RQN decreases. And here's the critical takeaway. So if the method has been properly developed and validated, you can still extract meaningful insights from samples with RQN values below 8. And as you can see, this is especially evident with the gene number 4 here, which shows increased expression at lower RQN levels. This is likely indicating a true patient-specific biological response. So what this tells us is that a robust method helps you to account for degradation and still detect real biological signals. But without all of this, without that rigority, low RQN quality can lead to false or misleading conclusions, particularly in clinical and regulatory settings. And this is why we have put such strong emphasis on quality control and method development, so that the data that we deliver is not only technically sound, but also biologically meaningful. For some take-home messages that will help you forward is to make sure that the sampling and shipping are optimized with clear instructions, especially for the mixing and freezing timing. And monitor your RQN variability between extractions to keep the data reliable. And if you find a variable appears, standardize the workflow further reduces. And these steps help you to ensure sample quality and consistency for trustworthy results. And with that, I will thank Kalle for the presentation.
All right, thank you so much. We are in our live Q&A. I'll just repeat those instructions that I just gave. So if you would like to ask a question at this time, please go to the Ask a Question box located at the bottom of your screen. And we do see some questions coming through. And we are having some technical issues with Dr. Günther's camera, but he is on the audio if you'd like to say hello there. And Dr. Burmeister, you may come off of mute and turn your camera on. Perfect. Thank you so much. And Dr. Günther, hopefully you can hear me. I'm going to go ahead and start with our questions. Our first question asks, can DNA be isolated from PAX gene blood RNA tubes? And Dr. Günther, let me know if you can hear me on the audio. Again, we apologize for the technical issues we're experiencing today. Okay, I'm going to move on. We do have quite a few questions that have come through. I'm getting some feedback that the audio is low. Are you able to hear me, Dr. Burmeister?
Yeah, I can hear it perfectly.
Okay, so of the questions that have come through, if you would let me know number-wise, which is relevant to your presentation. Dr. Günther is unable to hear on the audio as well, so I just want to make sure that I'm asking questions that will be for you and not for Dr. Günther at this time.
Right. I think most of the questions are in technical relative to the packaging tubes. We have got one question that is number 10, I think.
Okay, I will read number 10. All right, and that question is, what is your turnaround times? How many samples can you extract?
Yeah, and that of course depends on the complexity of the samples and the assay. But if you take in considering the packaging blood tubes, we have two different systems, automated systems, and we can extract up to a few hundred samples per day if they are running continuously. I think each system can take around 24 samples for each rack, and then there is 3 different racks for each set. So that takes about 2 hours per set. So it's about 250 samples per day that we can process.
All right, fantastic. Thank you so much. Checking in, Dr. Günther, if you're able to hear us, go ahead and say hello. Just want to make sure. Or perhaps you're trying to unmute. All right. So we do have a few additional questions that have come through. And Dr. Burmeister, these are relevant to Dr. Günther's presentation. Is that correct?
Yeah, I think most of the questions are technical questions regarding the vaccine and the stability and so on.
All right, so what we'll do is we will go ahead and make sure that Dr. Günther does have access to those questions following this webinar so that he can follow up directly. Unfortunately, again, we did experience some technical issues here today, so we appreciate your patience and sticking with us for that. We would like to thank you for your time and thank our presenters for their important research and their delivery today. We would also like to thank Labroots and our sponsor, Pre-Analytics, for underwriting today's educational webcast. Before you go, I would like to thank you again for joining us and for your questions. Again, those questions that we did not get to answer today and those that are submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. Additionally, the webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, I again thank you for joining us, and we look forward to seeing you again. Take care, everyone. Bye-bye.